I would like to start off by thanking the 2020 SU Undergraduate Research Symposium for inviting me to present my work. I'll be presenting on the protective effects of a parenting intervention on mother-child immune cell gene expression, the attached randomized control trial. Toxic stress such as domestic violence predicts poor developmental outcomes, poor maternal health, and risk for inflammatory-based disease. Domestic violence occurs in 4% of childbearing families in Canada, and this prevalence rate is likely an underestimation due to underreporting. Interventions that improve mother-child relationship quality can buffer the developmental and mental impacts of toxic stress. However, the impact of such parenting interventions on physical outcomes, such as immune system activity, is less studied, creating a knowledge gap in literature. Toxic stress, as mentioned before, increases the risk for inflammatory-based disease because toxic stress can get under the skin to affect immune outcomes through neuroendocrine pathways and by changing behavior. The conserved transcriptional response to adversity, CTRA, is a stress-linked immune cell gene expression profile. Diverse forms of adversity, including domestic violence, evoke CTRA, which is characterized by increased pro-inflammatory signaling and decreased antiviral signaling. Higher CTRA is associated with inflammatory-based diseases, such as coronary heart disease. The objective of our study was to determine whether a parenting intervention predicts healthier immune cell gene expression profiles, which are characterized by lower pro-inflammatory and higher antiviral gene expression, in a sample of mother-child dyads affected by domestic violence. Our sample consisted of 20 mother-child dyads recruited from a Discovery House, which is a domestic violence shelter in Calgary. Then a randomized control trial was done of the attached parenting intervention. 10 dyads were randomly assigned to the intervention group and 10 to the control waitlist group. The attached intervention is a 10-week psychoeducational program that fosters maternal reflective function, where the skills a mother needs to understand and respond appropriately to her child's mental states. The attached parenting intervention has 3 components. The first is a 3 to 5 free play session that is videotaped. Second, the mothers are asked to pick a day-to-day real-life stressful situation. And third, a hypothetical situation that is mildly stressful. In all 3 of these components, the mom talks through her own and her child's thoughts, feelings, mental states, and intentions. The goal is to develop this reflective function scale in the mothers because reflective function allows for parents to appropriately regulate their own and their child's feelings and behavior. Therefore, it is an essential scale for mothers to respond more sensitively to her children, which fosters secure attachment and healthy relationship development. Our outcome variable, immune system activity, was captured by CTRA. In order to do this, we collected dry blood spots from mothers and children in both groups following the intervention period, conducted mRNA sequencing, and assessed for CTRA gene expression. Finally, mixed effect linear model analysis of 53 CTRA indicator genes was conducted. In terms of results, significant differences were found in CTRA scores between the intervention and control group. Mothers and children in the intervention group had lower and healthier CTRA scores, meaning that they had lower pro-inflammatory and higher antiviral gene expression post-intervention compared to the moms and children in the control group, as illustrated in Figure 1. Table 1 and 2 show the child and maternal sample characteristics, respectively. Although the sample characteristics differed between the intervention and control group, differences in CTRA scores between the 2 groups still persisted even when we controlled for variables such as age and race. In conclusion, mother-child dyads who participated in the attached program had healthier immune cell gene expression profiles post-intervention compared to waitlist controls. Parenting interventions similar to attach could decrease the impact of toxic stress on maternal-child immune system activity. These findings have important implications for protecting the health of mothers and children living in adverse circumstances and will inform the future use of the attach intervention. I would like to acknowledge all these organizations. I want to thank Alberta Innovates for funding my project, and I want to extend my sincerest gratitude to Dr. Kara Ross for being an amazing supervisor. Thank you for your time.